everyone, this is Dr. Nika Taloma and this is a lecture video on the Pentose Phosphate pathway. Currently, I am on facility quarantine because of an exposure to a positive case, so I thought of recording these lectures instead in order for it to be ready. So when we talk about Pentose Phosphate pathway class, this is also known as the hexose monophosphate shunt or pathway, or the phosphogluconate pathway. And when we talk about the pentose phosphate pathway, it is anabolic in nature. So if we try to differentiate what is anabolic to that of catabolic, pag sinabi nating anabolic, it means to build, catabolic means to break down. So this is an anabolic reaction in its nature. So what are the pentoses and why do we need them? So we have the DNA and RNA. So we're going to discuss cofactors in enzymes. And these pentoses, where do we get them? So we can get this from our diet and from glucose and also from other sugars. So this is via the pentose phosphate pathway. So this pentose phosphate pathway, I want you to remember that it has two phases, an oxidative phase and a non-oxidative phase. So you will know later that this oxidative phase is irreversible in nature, while the non-oxidative phase is reversible. So here, we can see, as I've mentioned earlier, the pentose phosphate pathway is also known as the hexose monophosphate shunt. This is its other name. And when we are trying to understand a pathway class, we always remember to ask, what is it for? So when we had the review for the medical board exam, ito yung tinanong sa amin. And it was difficult for us to answer because when we tried to study on the different pathways, we did not ask this question. Ito pala yung question na dapat na tinatanong natin. So para saan ang pentose phosphate pathway? So it produces an ADPH. Also, it produces ribose 5-phosphate, which is required for biosynthesis nucleotides. So, ito yung makailangan natin malaman. What are those for? Para saan ang NADPH? And what is a ribose 5-phosphate for? So, they provide a mechanism for the metabolic use, also of 5-carbon sugar. And it neither produces nor consumes adenosine triphosphate. So this pentose phosphate pathway, this is a pathway that provides a major portion of the cell's NADPH, as we have mentioned. And this NADPH functions as a biochemical reductant. So we will discuss more of that later. So as I've mentioned, pag tinanong kayo, if you will be asked in the exam, does it produce or consume ATP? So this pathway does not. So these are the common questions that are being asked, class. So here, interconversion reactions of a hexose monophosphate pathway can function in several different directions. So yung interconversion, we will understand that more later. And rate and direction of the reactions at any given time are determined by the supply of and demand for intermediates in the cycle. So this is true for all pathways, class. It will depend on the presence of your intermediates that are being used in the cycle. Okay, so in our next slide, we have here, where does it occur? So that is the next question that you would want to understand. Where does your pentose phosphate pathway occur? So in the specific part of the cell, we have to answer that. And the answer, class, is in the cytosol of the cell. In the cytosol. But you have to remember that it is particularly important in the liver and other mammary glands which are active in the biosynthesis of fatty acids. So red blood cells as well, and tissues that produce like lipids or fats. So ano ba yung mga tissues or organs that produces lipids? We have the liver, the adipose tissue, adrenals, thyroid, testis, and lactating mammary. Remember this, class, it's not just specifically the mammary gland, but it is more specific to a lactating mammary gland, okay? So, to a lactating mammary gland. So, where does it occur in the cytosol? So next, we also need to know what is the substrate, because sabi nga natin, we will be able to determine or the product will be determined by the intermediate, or the how fast this pathway occurs will determine on the intermediates in the process. So what is the substrate? So kailangan natin malaman the substrate is glucose 6 phosphate. Okay, glucose 6 phosphate. So in the glucose 6 phosphate, maalala nyo, this is important also for glycolysis. So next, what are the important products? We've mentioned this in the second slide. We have NADPH and ribose 5 phosphate. Okay. Now, another thing that you have to ask in every pathway is, what is the rate limiting step? And when we talk about rate limiting step, this is the slowest step in the process. Meaning, kaya siya rate limiting because it will depend on how slow that step is. Okay, and here, the rate limiting step is the conversion of glucose 6 phosphate to 6 phosphogluconate here. Okay? So it is in the oxidative phase, and the enzyme is your glucose 6 phosphate dehydrogenase. Okay, so we have to take note of this because it's important for us to understand that the rate limiting step is the step that causes or that identifies how fast a certain process is. Okay, so this is usually asked in the board exam. Okay, so as I mentioned earlier, there are two phases. Non-oxidative phase and an oxidative phase. So as we can see it here. So in the oxidative phase, this is the portion that is included in that phase. And then in an oxidative phase, we can see here that there are different reactions. So we move on to the next slide to at least help us understand it better. So here, so we have two phases. What occurs first is the oxidative phase, followed by the non-oxidative phase. So I want you to remember that. So it's the oxidative phase first that occurs. Now, ang mnemonic ko here when I was reviewing for the boards is OI. OI. The oxidative phase is the one that is irreversible, while the non-oxidative phase is the one that is reversible. So the key enzyme in the oxidative phase is glucose 6 phosphate dehydrogenase. And if you remember, this is the enzyme in the rate limiting steps. So what are the products? So this table is very important. It's the summary of the pentose phosphate pathway. So here there are different products. For the first phase, we have 2 NADPH. And we have ribulose 5 phosphate or ribulose 5 phosphate. Okay? Now we proceed to the next phase. We have the second phase, which is the non-oxidative phase, which I mentioned is reversible. Before I forget, this first phase, it consists of 3 reactions, which we are going to talk about later on. So here in the second phase, we have the key enzymes. We have transketolysis with a cofactor, which is thiamine. So when we talk about cofactor, these are non-proteins that helps in the biochemical reaction. So what are the products of our non-oxidative phase? We have ribose 5 phosphate, fructose 6 phosphate, glyceraldehyde phosphate, and other carbohydrates. So this table class is very important for you to understand and remember. Okay? Parang this is a summary table. So now let's move on and discuss the specific processes. So this one, which is the summary of the pentose phosphate pathway, we are going to dissect the different parts or the different reactions that is inside this pathway. Okay? So first, let's talk about oxidative phase. Okay? When we talk about oxidative phase class, as I mentioned earlier, it is composed of 3 reactions. And the first one is the dehydrogenation of glucose 6 phosphate, which is catalyzes an irreversible oxidation of glucose 6 phosphate, okay, glucose 6 phosphate to 6 phosphogluconolactone. Okay, this is in a reaction that is specific for NADP as coenzyme. So the coenzyme is NADP, and this is the enzyme glucose 6 phosphate dehydrogenase, and this is the rate limiting step. The first reaction is the rate limiting step. So if we try to review that class again, the first step of the oxidative phase is glucose 6-phosphate, uses glucose 6-phosphate dehydrogenase as its enzyme. So this is the first enzymatic step. It converts NADP plus to NADPH. Remember, we've mentioned earlier that the product of pentose phosphate pathway oxidative phase is NADPH. So this is the first NADPH product. It is produced in the first reaction. So here, the mechanism is oxidation reaction of the C1 position. Hydride transfer to the NADP plus, which forms now a lactone, which is an intramolecular ester. And this reaction class is very favorable in the forward direction for producing NADPH. Okay, so what do we need to remember? This is the first reaction, and in the first reaction, this is the rate limiting step wherein we produce the first NADPH. Okay. Now, what we just discussed is this portion class, early portion here. This is the first reaction, okay? Now, let's proceed on to the next reaction. Here, before we proceed, I just would like to give a few descriptions on what is NADPH versus NADH. So this is a potent competitive inhibitor class. NADPH is a potent competitive inhibitor of the enzyme G6PD. And under most metabolic conditions, the ration of NADPH/NADP is sufficiently high to inhibit enzyme activity substantially. Ngayon class, if there is a increased demand of NADPH, this ratio of the NADPH and NADP decreases and the flux through the cycle increases in response to enhanced G6PD, the activity of G6PD. Okay, so that is the first reaction. Now, let's move on to the second part of the reaction or second reaction in the oxidative phase. Remember, ang pinag-usapan pa rin natin is the oxidative phase, which is an irreversible reaction. So, the enzyme here is lactonase. Okay, and what happens is the hydrolysis of 6-phosphogluconolactone. So, this will undergo hydrolysis. So, 6-phosphogluconolactone is hydrolyzed by lactonase. Okay, and 6-phosphogluconolactonase hydrolase. So this is irreversible, but it is not the rate limiting step. So it will undergo hydrolysis. Now, the mechanism that occurs is the lactone in 6-phosphogluconolactone is opened by hydrolysis, which is the addition of water to cleave a bond, usually a type of amide or ester. In this case, since the lactone is intramolecular, then 6-phosphogluconolactone is opened up to the acid form of gluconate. So this, through hydrolysis, this is opened up, okay, to form the acid form, which is the 6-phosphogluconate. And often these types of hydrolysis reactions are very favorable. Okay, so nagkaroon tayo ng hydrolysis which opened up 6-phosphogluconolactone, which now caused the formation of the acid form, which is 6-phosphogluconate. Okay, this is the second portion. Now, let's move on to the next reaction. So in this reaction class, we have the third reaction, which is the formation of ribulose phosphate. Okay, so here, subsequent oxidative decarboxylation of 6-phosphogluconate is catalyzed by 6-phosphogluconate dehydrogenase. Okay? So, irreversible din ito. Sabi nga natin, this is the third reaction of the oxidative phase. Therefore, it is irreversible. So, it produces class ribulose 5-phosphate. Yes. It also produces NADPH, which is the second NADPH, and carbon dioxide. So, ito yung third reaction natin in the oxidative phase. So, the enzyme is 6-phosphogluconate dehydrogenase, which now performs oxidative decarboxylation, which converts the 6-carbon skeleton into a pentose. So 6-carbon skeleton into a pentose. So the mechanism is the C1 carboxylate is removed as the C3 position is oxidized to a ketone, which now makes a 5-carbon ketose ribulose. So the terms are sometimes confusing, class, but what I want you to remember is that we are talking about the pentose phosphate pathway, which has 2 phases. First, the oxidative phase followed by the non-oxidative phase. In summary, the oxidative phase has 3 reactions. And the first reaction is the one that is the rate limiting in its step. But nevertheless, all 3 reactions are irreversible. Okay, now here, in this portion, we are going to discuss the enzyme Phosphopentose Isomerase. So here, the typical ketose to aldose conversion is seen. So this is another example of general acid-base catalyzed reaction wherein D-ribulose 5-phosphate is converted into D-ribose 5-phosphate by the enzyme Phosphopentose Isomerase. Okay? So dito natin nakikita yung production of D-ribose 5-phosphate. Now, having discussed the 3 reactions for our oxidative phase, we now move on to the non-oxidative phase. So the non-oxidative phase, this will catalyze the interconversion of 3, 4, 5, or 7 carbon sugars. So ito lang, class, what we want to take note in non-oxidative phase is that it can recycle pentose phosphates back to glucose 6-phosphates. So for example, mas kailangan pala ng body, the cell needs much more NADPH than pentose, diba? Kanina na-produce yung ribose 5-phosphate. So what if the cell needs NADPH more than that of pentose? In the non-oxidative phase, the D-ribose 5-phosphate can be converted back to glucose 6-phosphate in different enzyme catalyze steps, multiple enzyme catalyze steps. So what we're trying to say here is we can recycle the pentoses back to glucose 6-phosphate if there is a need for more NADPH than that of pentose. So the recycling of 5 carbon skeletons as expected for stoichiometry below is an example. Now, in this conversion, it will permit, conversion of ribulose 5-phosphate back to ribose 5-phosphate, which is needed for nucleotide synthesis, and also intermediates of glycolysis. So ribulose 5-phosphate back to intermediates of glycolysis. For example, we have fructose 6-phosphate and glyceraldehyde 3-phosphate. And this is controlled primarily, pati itong non-oxidative phase is controlled primarily by the availability of intermediates. Now, so take note, class, ang coenzyme natin for the non-oxidative phase is thiamine pyrophosphate. Now, I want you to take note here. At the end of the oxidative phase, class, what were we able to produce is the ribose 5-phosphate, tama? And our substrate is glucose 6-phosphate. So we can see here, this is the oxidative reaction of the pentose phosphate pathway. This is the first one we discussed earlier. This is the irreversible phase and this is the first phase. So from the substrate of glucose 6-phosphate, ginawa niyang ribose 5-phosphate. And this process is irreversible. Once that you already converted it to ribose 5-phosphate, you cannot reverse it anymore using the oxidative reaction. But what can we do if we need more glucose 6-phosphate rather than ribose 5-phosphate? We can now use what we call the non-oxidative reaction. So in the non-oxidative reaction, class, we have here ribose 5-phosphate, and through multiple enzymatic catalysis steps or enzyme catalyze steps, we can bring it back to glucose 6-phosphate. So not through the oxidative reaction or oxidative phase, but through the non-oxidative phase. So here we have ribose 5-phosphate, and through an epimerase, it is converted into cellulose 5-phosphate. Through our transketolase class, it produces glyceraldehyde 3-phosphate, which is also an intermediate for glycolysis. So this is important for us to take note. Okay? So here, using transaldolase, it can also convert it to erythropose 4-phosphate, and through transketolase, convert it back to fructose 6-phosphate, and then through phosphohexose isomerase, it can be converted back to glucose 6-phosphate. So that is one enzyme catalysis step. Another is from ribose 5-phosphate, it will be converted by transketolase to sedoheptulose 7-phosphate, and then through transaldolases, it could be converted to fructose 6-phosphate, which is again an intermediate of our glycolysis, and then back to a glucose 6-phosphate. Okay, so these are different enzyme catalysis steps class that helps us convert back pentose into glucose 6-phosphate if the body needs it more. Okay, in this next slide, we can see here fructose 6-phosphate has been emphasized. So here, it only means that the conversion of pentose phosphates to intermediates of glycolysis, which are glyceraldehyde 3-phosphate, okay, fructose 6-phosphate, and they are being converted using the transketolase and transaldolase. Okay, so in the next slide, we see here, on the other hand, for example, class, if the condition demands for pentoses for incorporation to nucleotides and nucleic acid is greater than the need for NADPH, we have here the fructose 6-phosphate. So non-oxidative reactions can provide biosynthesis of ribose 5-phosphate from fructose 6-phosphate in the absence of oxidative steps. So this is just an emphasis that it could also be through the fructose 6-phosphate. So even if the oxidative reaction is irreversible, we have the non-oxidative reaction class that now converts back ribose 5-phosphate to glucose 6-phosphate if there is a need for more NADPH. Okay? Now, here in this next portion where we are showing the glycolysis, we would like to emphasize here, class, that hexamonophosphate shunt is not an isolated repetitive cycle, but rather it is integrated with that of glycolysis. So remember here, this is the process of glycolysis. We can see here fructose 6-phosphate, glyceraldehyde 3-phosphate molecules, which are intermediates of glycolysis and also serves a part in the hexose monophosphate shunt. Okay? So, hindi siya isolated process. Rather, it is integrated also in glycolysis. So, that are required to transform pentoses, so ribose 5-phosphate, back to glucose. Okay, back to glucose. So glucose